Hey Ava, what are you up to?
I'm just looking
Is that the article about tumor irradiation in mice? I started reading that earlier, but I didn't get a chance to finish it. Aren't the researchers trying to determine if using high doses of radiation at a really fast rate would have positive effects on the growth of tumors?
Yeah, I just finished reading it. It was fascinating. The scientists hypothesized that using this new flash technology, which is actually just a cool laser-powered device, would allow the doctors to use higher radiation doses to treat cancer without harming the patient's normal tissues too much.
That's so cool.
So cool.
Wait, were you guys talking about the irradiation in mice? I was just reading that. Tell me more.
Okay, I think I know what I'm talking about now, so let me just break it down for you guys. So in this specific... Wait a second. This isn't right. I need my article reading goggles. Okay, much better. So, in this specific study, researchers used a compact laser-powered device to generate high-energy protons greater than 60 million electrovolts and conducted an experiment on mice models with human tumors. They were able to control the radiation dose delivered to the tumors using the laser-powered device as well as the clinical proton source and looked at how the radiation affected the tumor growth.
Wait, are you guys talking about flash? I was just reading about it. Let me tell you more. So flash is a way of delivering radiation very quickly, in less than 1/10 of a second, at much higher rates than what is typically used in regular radiation therapy, which takes minutes. This fast radiation delivery has shown promise in treating cancer because it seems to work well on tumors, while causing less damage to healthy tissues. This could potentially allow doctors to use high radiation doses to treat cancer without harming the patient's normal tissues too much.
However, before flash radiation therapy can be used as a standard treatment, studies must be conducted on how it affects both cancerous and healthy tissues. We need to understand various factors like how much radiation is delivered in each quick burst, how often these bursts happen, and how long the entire treatment takes.
So I did get a chance to read a bit about it, and I think I understand it to a pretty good degree, but I think these would help. Thank you. So when conducting these experiments, the researchers had to rely on small equipment called research accelerators, which are used on experiments on small animals. These accelerators can deliver radiation in different ways and at different speeds, ranging from extremely fast, like milliseconds, to whole minutes. This helps the scientists understand how the radiation interacts with tissues at the different speeds. In this particular experiment, the Draco PW laser is used to target a special material, and this process generates protons. And the protons have a range of energies, and we can select a specific group of them to target. And these selected protons are then guided through a system using magnets and other devices. Very techy. One of these devices, called the ESA, helps shape the proton's energy spectrum, while others help make the proton beam spread out evenly. The protons pass through monitors and measurement devices in the air to ensure that they are delivering the correct dose. And finally, those protons are then delivered to a specific area within a mouse bedding unit where they deliver the intended dose of radiation.
Oh, now I get it. Okay, and the researchers also used a small animal model with tiny spherical tumors grown on a mouse's ear, which allows them to study how different types of radiation affects these small tumors. To ensure precise radiation delivery, they defined a target area, PTV, within the tumors and prescribed a specific radiation dose to this area. The prescribed dose had to meet certain criteria. It needed to be delivered within a short time frame, no longer than 4 minutes, and with a specific energy spectrum. This energy spectrum required the protons to have a particular range of energies to ensure the radiation penetrated the tumors effectively while minimizing damage to the surrounding healthy tissue. To achieve this, the researchers used the Draco PW laser to generate protons. These protons were then directed through the complex system of magnets and scatters to shape their energy spectrum and ensure they hit the PTV accurately. They also developed a monitoring system using a time-of-flight spectrometer to track the proton energy and make sure it matched their desired spectrum. To deliver the prescribed dose, they used a multi-shot approach, combining several individual shots of protons to reach the required dose level. The laser energy was adjusted to control the dose for each shot. They monitored the dose with specialized instruments such as transmission ionization chamber, and dosimetry radiochromic film.
Aw, mice are so cute. But a total of 61 mice in the experiment and divided them into different groups. Some received proton radiation, some were given fake treatments with no radiation, and some were irradiated with x-rays for comparison. Additionally, there were control groups of mice that didn't receive any treatment at all. The sham-treated groups underwent all the same procedures as the groups receiving radiation, except they didn't actually receive radiation. These sham-treated groups were important to understand if any effects on tumor growth were due to the experimental procedures rather than the radiation itself.
Wow, this is a lot of information. I need to take it in. Okay, I'm ready to hear what they discovered.
Now that she's done with her nap, I can tell you guys the results. But first, always. So, when we compare these results of the mice that weren't irradiated or were sham irradiated, the control, we can see a clear effect caused by the radiation. In some cases, the tumor growth was significantly delayed or even controlled by the radiation. However, these cases were relatively rare and only happened a few times in the LPA proton group. The observation is interesting, and it motivates further research to determine how effective LPA proton radiation is at delaying tumor growth, but more data is needed to draw strong conclusions.
They actually discovered more than just that, but first, thank you. Better. Since this was a smaller study with a limited number of mice, only 61, the results based on X-ray data that were collected separately were not adjusted. However, in any future larger studies, this should be accounted for to account for any variations in how the mice respond to the radiation. The researchers did not find any evidence that environmental conditions or experimental procedures influence the results, which means no confounding variables. This is important because it means that future studies can rely on consistent conditions and therefore can be replicated. To reach a more definitive conclusion about the differences between the laser-driven and conventional proton radiation, a much larger study should be conducted with at least 300 mice in total. This would give us a better understanding on how effective each type of radiation is at delaying tumor growth.
Okay, this is crazy. I did not know how powerful lasers were. This seems to really set a standard and foundation for new scientific discovery. Do you guys know, what they're gonna do in the future with this information?
Yeah, as a matter of fact, beyond this study, this platform can be used as a reference to deliver customized radiation doses to a wide range of samples, allowing us to study how radiation affects different materials or organisms. We can even deliver very high doses of radiation in an extremely short time, which could be used for exciting future applications like investigating the effects of high-speed radiation on biological organisms or even exploring radiochemistry.
So, I think scientists did a good job. They took confounding factors into account and found some interesting information that could be applied to future use. However, I do think that in the future, they could use a larger sample group for more accurate results, and they could also test it on different organisms so that we can generalize the results to a more broad population.
Wow, who knew what you could do with some mice and lasers?
Right? I am so glad that we all got the chance to come together and chat about this. Maybe we could, same time next week, analyze another research paper.
I'm so excited.
Awesome.
Same time next week?
For sure.